So in today's lecture, we will be going to be start off with the operons and how the operons and their system and the mechanism is going to worked out. So whenever the complete procedures are going on, or whenever the system of regulatory activities are going on, at that point of the time, there are some things which are happening and there are some things which are working on and working off. So these conditions which where the system is going to be in a turn on condition and in the turn off condition is how the complete mechanism is being regulated. So whenever some systems or some things are getting regulated or some procedures are happening, and some sort of the procedures are there, some things are there which are showcasing the ability to perform some sort of regulatory activities, and these regulatory activities showcasing their complete sort of entire thing, like their complete regulatory thing, like working out and done their procedures, and then entirely like completing the whole mechanism of how all these things are togetherly worked out and how these things function together. So this thing or this mechanism where cluster of genes are being regulated by a single promoter system. Okay, so here if you could see in this diagrammatic representation, so here there are a lot of genes which are there. Okay, now no matter what, how many genes are there, no matter what, either there are 2 genes or 3 genes or multiple genes are there, but all these genes, all the complete system of these genes are being regulated by a single promoter. Okay, so this is the cluster of the genes, no matter how many genes are there in this complete mechanism and in this complete system, but all these genes are being regulated by a single promoter. So here this entire thing where this cluster is being regulated by a single promoter, it is going to mediate its activities responsible for carrying out the formation of the mRNA, that is the transcription procedure, and then from this mRNA it will be going to entirely form the proteins from these genes, okay, which will ultimately helping out and carrying out the complete regulatory procedures. So here the proteins are getting formed from this mRNA with the help of translation procedure. So this is how the complete formation of proteins is going to take place, and the way the proteins are formed, whether it could have been a switch on mechanism or whether it could have been a switch off mechanism, all these entire things and the procedures and everything related to all these mechanisms is the operon system. So how these genes are regulated, how the complete mechanisms are being regulated in this complete operons, that we will be going to study of in today's lecture. So basically there are 2 major categories in which operons are being divided. So these 2 categories are nothing but the catabolite, that is the degradative system, which example is given by the Lac, Ara and Gal operons, and also the anabolic, that is the biosynthetic system, where its examples is given by the tryptophan operon, right? So here there are other terms and terminologies which are also there, essential factors and features in case of these operon systems of mechanism. So here these regulatory mechanisms are being majorly divided into 2 major categories, right? So these 2 major categories would have been the negative regulation and the positive regulation, and how these entire regulatory mechanisms will be going to worked out, and then helping the entire regulation of gene expression to carry out a particular procedure, to carry out certain systems of procedures. Now here in this case, the term operon is being described and derived by the scientist Jacob and Monod first time in Escherichia coli in 1961. In the year 1961, he has discovered this operon system of mechanism in the bacterial system and concluded, or sorry, um, yeah. Okay, so the first time this scientist Jacob and Monod discovered this operon things and everything in the E. coli in 1961, and then he discovered that the complete mechanism of regulation are being under the control of a single promoter system, and here these operons and these things will be going to helping out and working in case of prokaryotes, as it would have been very difficult for the entire mechanism in the eukaryotes to cope up with all these things related to the complete mechanisms and everything. So that's why the operon things are present only in case of prokaryotes with some exceptional eukaryotic mechanisms. So now in detail we will be going to study of the things and the major activities related to the lac operon or the lactose operon, which is the first described or discovered operon by the scientists. So this is the structure of the lactose, where the galactose is being attached with the glucose molecule. So yeah, so whenever we are studying a complete cell or a system or a mechanism, so at that point of the time, whenever the bacteria are growing on a culture plate or on a cultured medium, so at that point of the time, what will be going to happen when the complete growth medium is being depleted in case of the bacterial cells or the system? So consider a plate is there, a colony basically of bacteria which are growing, that is the E. coli. So in these, what is going to happen, as the bacteria are going, they need some sort of the energy source from their environment. Now consider the plate, only the plate, being the environment for the bacterial cells. Okay, the plate is the entire environment for the bacteria, so we need to put some sort of the energy for the bacteria to grow and carry out whatever processings they want to carry out, and their functioning whatever they want to carry out. So here in this case, what is going to happen, when the initial source for the bacteria's energy, that is the glucose, which is the simplest form of the sugar, which could have been utilized by these bacteria, is being depleted. So in this condition, what is going to happen? The bacteria initially stops their growth, not actually stops their growth, but their growth is being completely, like, it is paused for some sort of the time, or for some lag period of time. Now during this lag period of time, what these bacteria will be going to do after the glucose is being depleted from the complete environment, they will be going to turn on the enzymes which are necessary for carrying out the lactose operon, because since the glucose is being depleted, the bacteria needs the other sugar sources which are available in the environment, and if the lactose is available in the system, in the environment, then the bacteria would try to utilize these lactose sugars, and then carrying out, like, they utilize these lactose by carrying out the complete regulation mechanism of the utilization of lactose, that is carrying out the lac operon. Now to turn on this lac operon, they need some genes. The bacteria needs some gene to catabolize this lactose sugar, that is to break this lactose sugar down, so that it could get the galactose plus glucose, that is the simplest form of the sugar again, which could have been easily utilized by these bacterial cells. So here what they will be going to do, and here what the main thing they will do, is they will synthesize three types of genes, or three types of complete genes which are there, or enzymes which are necessary for this complete mechanism of lac operon. So which genes are these? These are basically encoded by these three three designations, that is lac Z, lac Y, and lac A, which is going to code for the enzyme. Firstly, lac Z is going to code for beta galactosidase, lac Y is going to code for galactoside permease, and lac A going to code for thiogalactoside transacetylase. Now all these three genes have various different types of the functions, like these beta galactoside is utilized to break this lactose down into galactose and glucose. The permease is utilized for the entry of the lactose into the complete cell membrane by creating the pores in the cell membrane. So cell membrane is there, the permease will be going to create some pores so that lactose could enter the complete cell membrane, and then later on carrying out its operon procedures. And the third one, that is the thiogalactoside transacetylase, is utilized for carrying out the acetylation procedure on the beta galactosidase. With the help of beta galactosidase, it will be going to add the acetyl group. But some scientists think that it functions other than this transacetylation procedure, so they usually told it as unknown factor or unknown things like that. Okay. So this is the general model of the operon system. Now here in this general model, the DNA is there, okay, double-stranded DNA is there, regulatory genes are there, which are entirely regulating this complete procedure. The operon system is there. Now here in this operon system, structural genes are there, the operator and the promoter. This promoter is going to regulate this entire operon system by carrying out the regulation on these structural genes. So structural genes, with the help of firstly transcription, will be going to form the mRNA, and that later on with the help of translation will be going to result out in the formation of various different types of the proteins, which are essential for carrying out various metabolic activities. All right. So now we'll have a look at the structure of Lactoperon. So this is the general structure of the Lactoperon, how the complete structural organization of Lactoperon is there, which is very important. Now here in this case of structure of Lactoperon, what is basically going to happen is, the DNA is there obviously. Now here in this case, 3 types of structural genes are there in case of Lactoperon. So here this Z is going to be encoded by beta galactosidase, as we have seen earlier, the Y with permease, and A with transacetylase. Operator is there, which is completing the entire regulatory sequence or the regulatory circuit, and this is the structural circuit or structural genes are there. Right. So here the operator is there for the lac genes, and also the regulatory gene is there for the repressor, and the promoter for the repressor is also there in this complete structural organization of Lactoperon. So here in this case, the components, the various components which are there in this complete system, these components are being utilized to carry out different types of processing, as we have seen in the tabular representation, like how the complete genes are being carrying out their different sort of functioning and everything. Okay, so this is like that only in the different format. Moving forward. Now here, how these complete system, or why the lactose operon is being turned on. So whenever so initially, whenever the bacteria are growing on any medium or any environment, so at that point of the time, the bacteria always prefer some simpler sugars, okay, some simple sugar for their energy purpose, to gain the energy from those sugar molecules, the carbohydrates basically. Bacteria would not directly going to go towards the complex sugar molecules like the lactose or the arabinose and everything, etc., etc. So the bacteria will always going to prefer the simplest sugar molecule like the glucose. Now, if in case the glucose is not available in the system, okay, if in case the glucose is not there, or if by some things, or if by some processing, the glucose is being totally exhausted, okay, totally exhausted from the environment of the bacteria. So at that point of the time, the only available sugar for the bacteria in the complete medium would have been the lactose, if in case you have added the lactose sugar, or if by some medium the lactose is available there. And so now what the bacteria will do here, the bacteria will go towards starting the operons of this lactose, and then utilizing the lactose by breaking it down, by catabolizing it, and carrying out its use as a sugar source. Now here in this case, the complete Lactoperon is going to be working out on only 2 conditions. Firstly, the lactose should have been available in this complete mechanism to work out. And then the second and the most important thing is that the glucose should not be there in this complete system. The glucose should be exhausted. The glucose should have been completely depleted from the environment. Now, if in case a little or minute concentration of lactose is also there, then only the if even if the minutest concentration of glucose is there in the complete system, the bacteria won't move towards any other operon. It will completely utilize the lactose or the glucose which is available, and until and unless the glucose is entirely depleted from the complete system, then only the bacteria will be going to move towards other sugar sources, whatever available for them, right? So here, the work of the inducer in this lacoperon. Now, this complete operons or these lacoperons are working out with the help of inducer molecules. Now, these inducers which are here are basically not actually the lactose molecule, because it could have been like a completely degraded or like completely broken down by this gal which is going to be synthesized at the basal level. So here the inducer which is utilized is the isomer of the lactose, that is the allolactose, which is glucose 1,6 galactose. Now here, two other sort of artificial or gratuitous inducers or free inducers are there, which are also being utilized or which could have been utilized, and these are nothing but TMG, that is thiomethyl galactoside, and IPTG, that is isopropyl galactoside. So this is how the complete general mechanism of this lac operon is going to work out. Is that clear to all of you?
Yes, ma'am.
Yes, everybody, is that clear to all of you?
Yes, ma'am.